Usually when we talk about a scientific diagnosis, there's this underlying expectation of absolute precision.
Right, like it's math or engineering.
Exactly. It's like engineering. Imagine you fall off a ladder, right? You go to the hospital, and they take an X-ray of your arm.
Ouch. Yeah.
Yeah. And the doctor slaps the image up on the light board, points to a jagged white line, and says, there it is. That's the fracture. It's incredibly binary.
It is. It's either broken or it's not. The evidence is right there in black and white, and that kind of clarity is deeply comforting to the human brain.
Oh, absolutely.
We naturally want to take the chaotic physical world and just sort of organize it.
Yeah.
We want things to be visible, neatly categorized, and placed into their proper little boxes with clear labels.
But then you step out of the sterile environment of a modern hospital.
Right.
And you wade into the world of ancient agriculture.
Oh, boy, yeah.
Specifically, the genetics of plants that have been cultivated for centuries, and suddenly that metaphorical X-ray machine just shattered.
Completely falls apart.
Right. The diagnostic landscape we're looking at isn't black and white at all. It is incredibly, wonderfully murky.
It is the absolute definition of diagnostic muddy waters.
Yeah.
I mean, the neat little boxes we've used for hundreds of years turn out to be completely wrong when you finally look at them under a modern molecular microscope.
Welcome to the deep dive. Today, we are bringing you straight into those muddy waters to explore a botanical mystery that honestly plays out like a scientific soap opera.
It really does.
We're looking at a subject that is globally revered, an agricultural product, a specific vine variety that is so prestigious, it is often referred to simply as the king of grapes.
And it commands absolute respect.
It does, and it produces a liquid that people completely obsess over.
Yeah.
But there's a massive landscape-altering secret hiding in plain sight in the vineyards of northwestern Italy.
A massive identity crisis.
Exactly. This famous grape, it is experiencing a severe identity crisis. Because as modern genomics has only recently proven, it is not actually one grape at all.
That is the crux of the entire mystery. You have hundreds of years of entrenched agricultural tradition, legally codified assumptions, historical texts, all pointing in one single direction.
Right.
And then within the last few years, modern DNA sequencing comes along and completely rewrites the entire narrative.
So here is our mission for today's deep dive. We're going to unpack what we're calling the Nebbiolo Identity Crisis.
That's ominous.
Well, it is a big deal. We're going to explore the three famous faces of this vine. Historically, these are known as Lampia, Michet, and Rosé.
Right, the classic trio.
We are going to look at the shocking DNA revelations from 2017 and 2022 that basically upended everything farmers thought they knew.
The hard science.
Exactly. We'll dive into the cellular mechanics of a bizarre paradox where a plant virus actually creates a superior agricultural product, which is just wild to me.
It's one of my favorite biological weirdnesses.
Same. And finally, we'll break down the highly unusual molecular chemistry that makes this specific plant a master of visual deception.
And just to set the parameters for where we're going with this, we are keeping our lens strictly focused on the biology, the history, and the hard science of the plant itself.
Yes, very important point.
We're diving into phenotypic plasticity, genomic mapping, anthocyanin chemistry, all the fun stuff.
Right, which means we are actively steering clear of the commercial side of this world. We aren't going to talk about specific producer names or, famous wine labels or how much a bottle costs at auction.
We don't need to. The pure science of the vine is more than complex enough to keep us completely occupied.
It really is. I mean, the sheer volume of scientific scrutiny this plant has been subjected to is staggering, yet it managed to keep its most fundamental secrets hidden until the 21st century.
Which is incredible when you think about how long humans have been working with it.
Okay, so let's start by laying the historical foundation. This grape, Nebbiolo, is not some new hybridized cultivar cooked up in a university laboratory in the 1990s, right?
Not at all. This is an ancient plant.
Historical documents explicitly place it in the Piedmont region of northwestern Italy way back in the 13th century.
Yeah, there are local records from the year 1266 that talk about Nebbiolo.
1266. And there are even agricultural historians who argue that Pliny the Elder was referencing an early ancestor of this exact vine in the 1st century.
Which is just staggering longevity for an agricultural product.
Right. And for as long as humans have been writing about it, they have complained about one specific thing.
That it's a nightmare to grow.
Yes. It is an incredibly finicky, difficult plant. Biologically speaking, it possesses extremely high phenotypic plasticity.
Let's break that term down because understanding phenotypic plasticity is really the key to unlocking everything we are going to discuss today.
Please do, because it sounds like a superpower.
It kind of is. So phenotype refers to the observable physical properties of an organism. How it looks, how it grows, its size, its color.
Okay, the physical traits.
Right. And plasticity refers to its ability to mold or change. So when a plant has high phenotypic plasticity, it means it mutates and adapts its physical expression incredibly rapidly based on the specific environment it's planted in.
So it's hyper-reactive.
Exactly. It reacts to the microcomposition of the soil, the ambient temperature, the amount of rainfall, the angle of the sun, and even the local pathogens.
Wow.
You could take two genetically identical cuttings of this vine, plant them on opposite sides of the exact same hill, and within a few generations, they might look and behave completely differently.
And because it's so reactive, farmers historically noticed that the vines in their fields didn't all look uniform.
Right, they saw variations.
Right. Over centuries of working the land, long before the discovery of DNA or cellular biology, these agriculturalists categorized this grape into three primary groups growing right alongside each other.
And they gave them names.
They called these three distinct expressions Lampia, Michet, and Rosé. Now, in agricultural terms, these were referred to as biotypes. If I'm understanding this correctly, a biotype is basically nature's version of a software fork, right? You have the exact same base code, the identical DNA, but minor localized mutations or environmental factors have activated different features over time.
That's a brilliant way to conceptualize it. In a pre-DNA world, farmers categorized plants entirely by morphology.
Which is just how things look.
Literally just studying the form and structure of things.
Yeah.
You look at the shape of the leaf, the size of the grape cluster, the color of the fruit.
Right.
A biotype, scientifically speaking, is a group of organisms that share an identical genetic constitution but exhibit distinct physical traits.
Okay.
So when these farmers in the 1800s and 1900s looked at their fields, they saw three distinct physical expressions. And based on the science of the time, they made a perfectly logical assumption.
That these are all the exact same genetic plant.
Exactly. They assume they're all Nebbiolo, they're just presenting a little differently.
It's like having a set of identical triplets. Let's say one becomes a bodybuilder, one becomes a marathon runner, and one just sits on the couch eating potato chips.
I like the couch potato one.
Right. But if you just looked at them at age 30, their morphology is totally different. The bodybuilder is huge, the runner is lean. You might not even think they're related based on just looking at them.
But if you swab their cheeks...
The DNA is identical, they just lived different lives that shaped their physical forms.
Precisely. And in this historical paradigm, Lampia was the baseline. It was the standard original vine that farmers considered the true expression of the grape.
The default setting.
Right. Then you had Michet. The Michet vines looked different. They produced much smaller, looser, and more compact clusters of grapes.
Okay.
The overall yield of fruit was much lower, but the resulting agricultural product was highly concentrated, rich, and intensely prized by the farmers.
So it made less fruit, but better fruit.
Exactly. And then there was the third triplet, Rosé. The Rosé biotype behaved entirely differently than the other two. It produced a visibly paler liquid, but one that was heavily perfumed, loaded with delicate floral aromatics. Plus, from a farming perspective, it possessed excellent drought and heat resistance.
Oh, so it was easier to grow in hot years.
Yeah, so for hundreds of years, you have this neat, tidy historical categorization. Lampia, Michet, and Rosé are all just three different outfits worn by the same ancient king.
It was a very comfortable assumption. It made sense of the physical world.
It did, until it didn't.
Right, because here is where the X-ray machine breaks. This neat historical categorization completely fell apart when 21st century genomic sequencing entered the picture.
The 2017 and 2022 studies.
Yes. Scientists decided to finally map the genome of these three biotypes to see what was actually going on under the hood.
And we need to emphasize that sequencing a grapevine is not like running a simple cheek swab through a commercial ancestry database.
Right, it's not a mail-in kit.
Not at all. It is a monumental computational task. Grapevine genomes are notoriously complex.
Why is that?
Well, they are highly heterozygous, which means they have a massive amount of genetic variation inherited from their parent plants. They're also prone to extreme structural rearrangements.
Like chunks of DNA moving around.
Exactly. Large chunks of DNA being duplicated, deleted, or just flipped around entirely.
So the 2022 study didn't just look at a few markers. They did what's called a de novo assembly using long-read sequencing and optical mapping.
Which is top-tier genetic mapping.
Right. Let's make sure that makes sense to everyone. If older sequencing technology was like trying to put together a 10,000-piece jigsaw puzzle by looking at the picture on the box and matching tiny individual pieces.
Which is hard enough on its own.
Exactly. A de novo assembly is like having to build that 10,000-piece puzzle without the box at all. You have no reference picture. You just have to figure out how the pieces naturally link together from scratch.
That's a great analogy, and the optical mapping takes it a step further.
How does that work?
Instead of just trying to read the DNA letter by letter, the A's, C's, T's, and G's optical mapping is like taking a high-resolution satellite image of the entire chromosome.
Oh, wow.
Yeah, it allows scientists to step back and look at the physical architecture of the genome, which allows them to map those massive structural variations accurately.
So they take this incredible cutting-edge technology and they point it at the 3 historic biotypes. And the very first thing they look at is Rosé.
And the results were an absolute bombshell.
I call this the Rosé revelation.
I like that. Because the data was unequivocal, and it completely shattered a centuries-old agricultural truth.
So the fundamental rule of cloning in agriculture, and remember, grapevines are propagated by taking cuttings of an existing vine and planting them, not by planting seeds.
Right, vegetative propagation.
Exactly. So the rule is that true clones of a parent grape should have identical DNA makeup. You are just creating a physical extension of the exact same plant.
Right.
When the geneticists map the alleles, which are the specific gene markers of these biotypes, they found that the standard Nebbiolo clones, like the Lampia vines, all possessed what are called AA alleles at specific crucial locations on the genome.
Yes, those AA alleles are essentially the biological fingerprint of the Nebbiolo variety.
Okay.
Every true clone, no matter how much its morphology has shifted due to phenotypic plasticity over the years, will still carry that AA fingerprint.
But then they look at the genome of the Rosé vine, the vine that has been growing side by side with Lampia for centuries, the vine that was historically classified as a direct sibling.
A third triplet.
Right. And it didn't have the AA alleles.
It did not.
It had AT alleles.
And the biological mechanism behind that discovery is profound. The presence of that T allele completely breaks the software fork analogy we used earlier.
It breaks the triplet analogy, too.
It proves, beyond any shadow of a doubt, that Rosé is not a clone. It is not a biotype. It is an entirely genetically distinct variety of plant.
Wait. If they propagate by taking cuttings, how does a totally different plant just show up in the middle of a vineyard and trick everyone for hundreds of years?
That comes down to the difference between vegetative cloning and sexual reproduction.
Okay.
As you said, farmers maintain a variety by cloning, by taking a physical cutting. But occasionally, vines do reproduce sexually.
Like with flowers and pollen.
Exactly. Pollen from one vine fertilizes the flower of another vine, and a seed drops to the ground. That seed grows into a completely new plant that contains a genetic mix of both parents.
Oh, I see.
At some point in history, likely hundreds of years ago, a spontaneous cross-pollination event happened in those hills. A seed grew. The resulting vine produced grapes that looked and tasted somewhat like the others, and a farmer, not knowing any better.
They just took a cutting of that new vine.
Yep, they took a cutting and started propagating it as if it were the original.
That is wild. So genetically speaking, what exactly is Rosé if it isn't Nebbiolo?
That T allele indicates that Rosé shares only 50% of its DNA with the primary Nebbiolo grape.
Only 50%.
Right. The other 50% comes from a completely different, as yet unidentified, parent variety. In fact, genetically, the presence of that specific allele structure makes the Rosé variety more closely related to grape families like Pinot or Muscat than it is to its supposed Nebbiolo siblings.
It is a literal imposter.
It really is.
It's a botanical cuckoo bird sitting in the nest. And the irony here is just spectacular.
How do you mean?
Think about the strict regional production rules that govern high-end agriculture in Europe. The legal frameworks in Piedmont are incredibly rigid.
Oh, absolutely.
The rules for making the world-famous red liquids of this specific area dictate that they must be made from 100% Nebbiolo grapes. By law, no blending with other varieties is permitted. None.
It's heavily regulated to protect the authenticity and the heritage of the product.
But rosé is legally permitted. It is explicitly written into the regulatory code as an accepted official biotype.
Because of history.
Exactly. It's an imposter that snuck past the guards, integrated itself into the royal family, and got grandfathered into the strictest legal codes in the world simply because it has grown there for so long and does such a great impression of the main grape.
It's a really good disguise.
And farmers historically loved it because it added these beautiful, delicate floral aromatics to the final product. And like you said, it survived hot summers better than the main vines, so they just kept planting it.
And this raises a really fascinating tension between science and tradition. The law codifies agricultural tradition, but that tradition was entirely based on a morphological misunderstanding.
Right. They didn't have DNA tests in the 1800s.
Exactly. The people who wrote the laws truly believed rosé was just a physical variation of the same plant.
Right.
Now that science has proven it is a genetically distinct variety, you have a legal paradox.
Oh, I hadn't thought about that.
Yeah, by strict genetic definition, any product containing the rosé grape is a blend, which violates the spirit of the 100% purity law. Yet, because it contributes so positively to the sensory profile, and because it is so deeply woven into the historical fabric of the region, the agricultural authorities have essentially embraced the anomaly.
They just shrugged and said, well, it's been here for 400 years. It tastes good. Let's just keep calling it a biotype. I love that.
Very pragmatic.
It is. So that solves the mystery of the third triplet. Rosé is a totally different plant, but what about the second triplet? What about Michet?
Michet.
This leads us to our next genomic shocker, and I have to admit, when I was reading the research on this, it completely broke my brain. Because this doesn't involve cross-pollination or genetic imposters, it involves a viral infection.
Yes. The reality of the Michet biotype is perhaps one of the most fascinating biological paradoxes in modern agriculture.
So let's look at the Michet paradox. The researchers driving the 2022 whole genome sequencing project took the primary assembly of the Michet vine, which is scientifically designated in the catalogs as clone CVT71, and they compared it directly, nucleotide by nucleotide, against the standard Lampia vine, designated as clone CVT185.
Now, given what they had just discovered about rosé, you can imagine the tension in the laboratory.
Oh, they must have been expecting fireworks.
They were likely expecting to find some level of genetic variation. Perhaps Michet was also a cross-pollinated offspring, or at the very least, they expected to find significant localized mutations.
Like the structural variations you mentioned earlier.
Exactly. Structural variations in the DNA that would scientifically explain why Michet vines produce smaller grape clusters and lower crop yields than Lampia.
But that's not what they found at all. The results were staggering in their absolute silence.
Total silence.
The sequence showed 0 structural genomic variations between the two, none. Down to the absolute molecular base pair level, Michet and Lampia are the exact same plant. They are identical.
Which brings us back to the fundamental question of morphology. If their DNA is an exact perfect match, what is causing the massive physical difference in how they grow?
The answer is disease.
Disease.
Michet is not a genetic mutation. Michet is nothing more than a Lampia vine that has been infected and biologically altered by a specific chronic pathogen called the grapevine fanleaf virus.
And this requires a massive paradigm shift in how we traditionally view plant diseases.
Because normally viruses are bad news.
Usually when we think of a virus infecting an agricultural crop, we think of devastation.
Right.
We think of blight, massive die-offs, ruined harvests, and economic ruin for the farmers.
Yeah, the very definition of a pathogen implies an organism that causes disease, detriment, and destruction.
Right. If a farmer hears they have a virus in their fields, they usually panic. But here is the bizarre reality. In this specific instance, the virus doesn't kill the plant.
So what does it do?
Instead, it systematically restricts the vine's reproductive output. It stunts the plant's growth, forcing the vine to produce those much smaller, looser, and far more concentrated clusters of fruit that farmers noticed centuries ago.
To really grasp the magnitude of this, we need to dive into the cellular mechanics of the fanleaf virus and the biological concept of vine stress.
Let's do it.
So the fanleaf virus is typically transmitted by microscopic nematodes, basically tiny worms living in the soil, right?
Yes, soil nematodes. They feed on the roots of the vine and inject the viral particles,
And once inside the plant, the virus moves through the plasmosmata.
which are the microscopic channels that connect plant cells.
Right. It effectively hijacks the plant's vascular tissue.
It gets into the plumbing.
Exactly. It compromises the phloem, the tissue responsible for transporting sugars and metabolic energy from the leaves down to the rest of the plant.
And this is where the concept of vine stress becomes crucial.
Because stress makes it better.
In high-end viticulture, lower yields of fruit almost universally equate to higher quality in the final product.
Counterintuitive, but true.
Think about a highly vigorous, healthy plant. It wants to reproduce, so it pumps out massive amounts of foliage and a huge abundance of fruit. The metabolic energy of that plant, the complex nutrients it pulls from the soil, the sugars it synthesizes through photosynthesis, all of that limited energy is diluted across a massive crop load.
So the resulting fruit is often watery, simple, and lacking in complex secondary metabolites.
Right, it's spread too thin.
Like trying to paint a massive house with only 1 gallon of paint, the color is going to be incredibly weak.
Perfect analogy. But when a vine is subjected to stress, whether that's from being planted in poor, rocky soil, suffering from a severe lack of water, or in this case, battling a viral infection, its biological survival mechanisms kick in.
Okay, so how does it react?
The family virus acts as a natural biological yield restrictor. Because the plant's internal plumbing is clogged and compromised by the virus, it physically cannot push enough energy outward to support a massive crop of fruit.
It's physically blocked.
Yes. The plant realizes it's under attack, so it shuts down non-essential production. It funnels whatever limited metabolic energy it has left into a much smaller, highly restricted amount of fruit.
I was trying to visualize exactly how this works, and the best analogy I could come up with is like a governor on a high-performance engine.
Oh, I like that.
The vine's root system is trying to pump the gas, pulling up water and nutrients, trying to build this massive canopy of leaves and huge clusters of grapes. But the family virus steps in and acts like a restrictor plate. It chokes the fuel line.
Exactly.
The plant can't distribute the energy it wants to, so it dumps all the horsepower into just a few tiny, hyper-concentrated clusters.
That is exactly the mechanism at play. And the result is that the diseased vine arguably produces a vastly superior, far more concentrated, and highly prized agricultural product.
It's doing the farmer's job for them.
Essentially, yes. The virus is doing the incredibly hard, labor-intensive work of pruning and yield management from the inside out.
It's an evolutionary loophole. The virus is a parasite, but it found a way to make itself indispensable to the human farmers.
Which is brilliant if you think about it from the virus's perspective.
Right. By infecting the plant and creating this highly concentrated, supercharged fruit, the farmers looked at the vine and said, wow, whatever this Michet plant is doing, it's making the best stuff on the hill. Let's take cuttings of this specific vine and plant more of it.
And so they propagated the disease.
Exactly. The farmers actively propagated and protected the diseased vine, ensuring the survival of the virus, completely unaware that they were farming a pathogen.
It is a profound example of a symbiotic relationship masquerading as pathology. The humans became the vector for the virus's survival, all because the disease created a highly desirable morphological trait.
So we've solved the identity crisis. The king of grapes is actually a triad.
A very complicated triad.
You have the original baseline Lampia, you have the viral superstar Michet, and you have the genetic imposter Rosé. But what connects them? Why are all three of these entities so revered? Because regardless of their genetics or their viral load, they all produce a final product that shares a highly unusual, incredibly specific chemical trait.
And it is a trait that actively, almost aggressively, deceives the human senses.
This is where the molecular biology of the plant intersects directly with human sensory perception, and it is a fascinating collision.
It really is. The scientific data outlines a massive contradiction in the physical product this plant creates.
Let's talk about the visual deception first.
Okay. When you look at the visual appearance of the liquid produced by Nebbiolo, it is a masterclass in deception. It looks incredibly light, pale, and visually delicate.
Yeah, if you pour it into a glass, it is almost translucent.
And very uniquely, it almost immediately begins to shift away from a vibrant ruby color and takes on a pale brick orange or light garnet hue, even when the product is very young.
Visually, it communicates fragility. Your brain looks at that pale, watery color and immediately prepares your palate for something light, thin, and easy drinking.
Right. Your expectations are set.
But then you actually take a sip, and it is an absolute shock to the nervous system.
Oh, completely.
It hits the palate with massive fiercely astringent tannins, and screaming high acidity. It is an incredibly powerful, deeply structured, heavy-hitting liquid.
The visual cue is a complete trap.
Imagine sitting down in a restaurant, someone hands you a glass of water that visually looks like a very weak, pale herbal tea.
You'd expect it to be soothing.
Exactly, but when it hits your tongue, it has the intense, aggressive, concentrated grip of a triple shot of black espresso. Your brain practically misfires trying to reconcile what your eyes just saw with what your mouth is physically feeling.
The sensory dissonance is profound. And the biological science behind this massive deception lies entirely in the specific chemistry of the plant's anthocyanins and polyphenols.
Let's break those terms down, starting with anthocyanins, because this is where the color trick happens. Anthocyanins are essentially the water-soluble pigments found in the skins of the grapes. When the grapes are crushed and the juice sits with the skins, these pigments dissolve into the liquid and give it its color.
Correct. But here's the critical detail. Not all anthocyanins are created equal at a molecular level.
Yeah.
The scientific literature rigorously characterizes Nebbiolo as a pionidin 3O-glucoside prevalent cultivar.
Okay, that's a mouthful. What does that mean?
It means that the primary coloring agents the plant synthesizes are pionidin and cyanin glycosides. In organic chemistry, we refer to these as dihydroxylated anthocyanins.
Okay, dihydroxylated. What does that actually mean for the liquid in the glass?
It comes down to molecular stability. The electron rings in dihydroxylated anthocyanins are highly exposed. They're incredibly reactive to oxygen.
So they're fragile.
Very. The key characteristic of these specific molecular structures is that they are fundamentally unstable. The very moment they are exposed to oxygen, which naturally happens during the fermentation and aging processes, they begin to oxidize and break down.
The color literally falls apart.
Exactly. The molecular bonds weaken, and the color pigment simply drops out of the liquid almost immediately.
So the plant is genetically programmed to build color molecules that are designed to fall apart.
That's fascinating.
Yeah.
And what it doesn't have is just as important as what it does have, right?
Yes.
The genomic studies note that Nebbiolo contains remarkably low levels of malvidin and petunidin.
Exactly. Malvidin and petunidin are tri-substituted anthocyanins.
Tri-substituted, so structurally different.
Without getting too deep into the organic chemistry, they have more methoxy groups attached to their molecular rings, which essentially acts like a protective shield against oxygen.
Oh, so they are much tougher.
They are highly stable. These are the specific molecules that give other famous grape varieties like Cabernet or Syrah their deep, dark, inky, opaque, purple-black colors that last for decades.
But Nebbiolo doesn't have those. Right. Because the Nebbiolo genome genetically lacks the pathways to produce high amounts of these stable pigments and relies almost entirely on the unstable pionidin. Its color is biologically destined to be pale, translucent, and to shift toward that brick orange hue very early in its lifespan.
So the pale color isn't a defect. I mean, if a farmer produces a liquid that looks pale and orange, it doesn't mean they did a bad job farming or failed to extract the color properly.
Emphatically, no. The pale color is a feature, not a flaw.
I love that.
It is the direct, unalterable result of the plant's specific genomic coding and its metabolic signaling. The plant is executing exactly what its DNA programs it to do.
Okay, so that explains the weak visual presentation. But where does the heavy-hitting reality come from?
The payload.
Right. If the color molecules are so inherently weak and unstable, why does the mouthfeel resemble a triple shot of espresso? Why is it so aggressive?
That is the role of the tannins, which are an entirely different class of complex chemical compounds known as polyphenols.
Polyphenols, okay.
Yeah. While the plant's genetic code severely shortchanges it on stable color pigments, it absolutely overcompensates on tannin production.
How do tannins actually work on a biological level? Like, why do they make my mouth feel like it's drying out?
It's a fascinating chemical reaction. Tannins are extracted primarily from the skins and the seeds of the grape during the crushing process.
Okay.
When you consume them, these polyphenolic compounds actively seek out and bind to the proteins in your saliva.
Oh, weird.
Yeah, they literally cross-link with the lubricating proteins in your mouth, causing them to precipitate or basically fall out of solution.
That sounds uncomfortable.
It is. That loss of lubrication creates the intense, drying physical friction, the astringency that you feel on your palate.
It's literally stripping the moisture coating off my tongue.
Exactly. And structurally, the tannins produced by Nebbiolo are massive and abundant. They provide an immense molecular scaffolding for the liquid.
So it's built like a tank on the inside.
Yes. This massive tannic structure is what gives the final product its famous, almost unparalleled aging potential.
Because the tannins act as a preservative.
Precisely. The color pigments may oxidize and fade away within a few years, but that immense tannic architecture allows the product to survive, evolve, and remain vibrant in a cellar for decades.
It is a biological paradox that completely contradicts the delicate visual appearance.
It's a botanical magic trick. A pale, watery-looking liquid that is chemically armed to the teeth.
So this naturally leads to the big evolutionary question: why? Why did this specific plant evolve this highly unusual, highly unstable chemical makeup?
It didn't happen in a vacuum, that's for sure.
To understand that, we have to look at how the plant interacts with its extreme physical environment. Which brings us to the concept of terroir and how it triggers that phenotypic plasticity we talked about earlier.
This is where the concept of terroir, which, let's be honest, is so often used as a vague romantic marketing term to sell expensive agricultural products, is proven as hard, measurable biological science.
Let's define it clearly.
Terroir is the idea that a crop reflects the specific geography, geology, and climate of the exact place it is grown.
Let's start with the name of the grape itself, because it is literally named after its environment.
The name is a huge clue.
The word Nebbiolo is derived from the Italian word nebbia, which translates to fog. And according to the historical and scientific data, this refers to two very distinct physical phenomena.
First, the weather.
Right. It refers to the heavy, thick autumn fogs that roll into the valleys and blanket the hills of the Piedmont region right around harvest time in October.
It's a very dramatic landscape. And second, it refers to a physical characteristic of the plant itself.
The wax, right.
Yes, a thick, foggy, whitish bloom, a waxy coating, scientifically known as pruina, that heavily coats the outside of the grape berries.
And both the environmental weather fog and the physical waxy fog on the grape are directly related to the plant's absolutely extreme, punishing growing cycle.
It is a grueling cycle.
The growing season for Nebbiolo is precariously long. It is notoriously one of the very first vine varieties to break bud and push out leaves in the early spring, usually right around early April.
Right, it wakes up very early.
And it is the absolute last to be harvested, often hanging on the vine deep into late October or even early November.
It's a huge window of time.
It requires a massive, agonizingly long amount of time connected to the vine to achieve full phenolic ripeness, meaning the development of those complex tannins and aromatics we just talked about.
And from a biological and agricultural standpoint, that extended exposure is incredibly dangerous. It is an evolutionary liability.
Because it's outside for so long.
Exactly. By breaking buds so early in April, the fragile new shoots are highly susceptible to devastating spring frosts, which can wipe out an entire year's crop overnight.
Boom, one cold night and you're done.
Yep, and by hanging on the vine deep into late October, the mature fruit is exposed to autumn rains, plunging temperatures, and massive outbreaks of fungal rot. It is a plant that lives perpetually on the razor's edge of agricultural disaster.
So how does it survive? Why hasn't it just died out over the centuries?
This brings us back to the plant's brilliant chemistry.
Right. To survive this long, perilous exposure to the elements, the genome of the Nebbiolo plant is packed with specific genes that trigger the massive overproduction of compounds called triterpenoids.
Triterpenoids are specialized, highly complex lipids. They're essentially the biological building blocks that the plant synthesizes to create that thick cuticular wax, the pruina or foggy bloom, that coats the outside of the berries.
It's biological armor.
Exactly. This wax serves a dual purpose.
What's the first one?
During the intense blistering heat of the peak Italian summer, this thick lipid layer prevents the moisture inside the fragile, thin-skinned berries from evaporating. It stops dehydration.
Oh, it seals the water in.
Yes. Then, when the weather turns and the damp, heavy autumn fogs roll in during October, that exact same waxy coating acts as a physical hydrophobic barrier.
It repels the water.
Right. It prevents fungal pathogens from taking hold and rotting the fruit. The plant evolved this massive, energy-intensive, waxy defense mechanism specifically to survive its extraordinarily long, punishing hang time in this specific geographical location.
And the environment isn't just about the weather, right? It's about the dirt.
Its soil is everything.
The geological data highlights that the soil requirements for this plant are incredibly strict, which further dictates its chemical output. The primary region in northwestern Italy where it thrives is defined by highly specific, ancient marine sedimentary soils left over from when the area was an ocean floor millions of years ago.
And the plant is so sensitive that it reacts to microscopic changes in that dirt.
This brings us back to the hyperreactivity of phenotypic plasticity.
We keep coming back to that,
Right.
yes.
When the vine is planted in what geologists call Tortonian soils, which are characterized by higher amounts of calcareous marls and loose sand.
The lighter soils.
Yes, the lighter soils. The resulting chemical output of the plant yields a product that is highly aromatic, elegant, perfumed, and floral.
Okay.
But if a farmer takes a cutting of that exact same vine and plants it just a few miles over, on a hill composed of Cerevallian soils, which are much older, poorer soils, dominated by compacted sandstone and limestone, the plant alters its expression entirely.
The exact same DNA.
The exact same DNA yields a product with intense, brooding, massive tannic structure and much darker fruit profiles.
Nebbiolo is arguably the most site-sensitive agricultural crop on the planet. It acts as a perfect, hypersensitive geographical lens. It literally translates the specific mineral and chemical composition of the soil directly into its biological output.
Which explains a massive mystery in the global agricultural world. Why this plant absolutely refuses to travel.
Oh, it hates traveling.
You look at a grape like Cabernet Sauvignon or Chardonnay. They originated in France, but humans have planted them everywhere, from the valleys of California to the deserts of Australia to the mountains of South America, and they generally succeed.
They adapt well.
Right. They maintain their core identity. They still taste like Cabernet and Chardonnay.
Okay.
But Nebbiolo, it is a biological homebody. It refuses to leave its house. It is incredibly stubborn. When agriculturists attempt to take cuttings of Nebbiolo out of these highly specific, geographically isolated microclimates in northwestern Italy and plant them somewhere else, even somewhere with a seemingly similar climate, the plant essentially panics on a cellular level.
It shorts out.
Because of its extreme phenotypic plasticity, its genetic expression wildly shifts, trying to adapt to the foreign soil and sun.
It tries to change its clothes, but puts them on backward.
Precisely. The transcriptomes associated with anthocyanin and polyphenol biosynthesis, the exact microscopic cellular mechanisms that dictate its fragile color, its floral aromatics, and its massive tannin balance. They modulate differently in response to the new environment.
So it survives, but...
The plant survives, but it loses its identity entirely. It stops tasting like Nebbiolo and starts tasting like a generic, unidentifiable, often flabby and unstructured red grape.
It's amazing when you step back and look at the whole picture. It's like the plant is perfectly, seamlessly adapted and locked into this one specific, tiny corner of the earth.
It's highly specialized.
The damp autumn fog, the specific marine sedimentary soils from millions of years ago, the terrifyingly long growing season, the triterpenoid wax armor it builds to survive, and even the local soil nematodes carrying the fan leaf viruses, they all lock together like microscopic puzzle pieces to create this one entirely unique chemical profile.
Take one piece out and it fails.
Exactly. If you take away just one piece of that puzzle, if you plant it in different dirt, or if you cure the virus, the whole picture falls apart and the magic disappears.
It is one of the most profound examples of gene-by-environment interaction we have. The plant is not just an isolated biological organism executing a static string of code. It is a living, active, highly sensitive reflection of its precise geographical and ecological coordinates.
Okay, let's take a breath, step back, and summarize the incredible journey we've taken through the muddy, diagnostic waters today.
It's been quite a ride.
We started by looking at this revered king of grapes, a plant with a global reputation for regal, singular perfection. And instead, by looking through the lens of modern genomics, we uncovered a wild botanical soap opera.
A fascinatingly messy, virus-laden, genetically confused reality.
Yes. We found a historic trio of supposed identical siblings that turned out to be anything but. We have the original Lampia, the baseline standard, carrying the true AA genetic fingerprint.
The anchor.
Right. Then we have Michet, which farmers thought was a unique, superior clone, but turned out to be nothing more than a Lampia vine that has its vascular system choked by a fan leaf virus.
A pathogen that paradoxically makes the resulting fruit infinitely more concentrated and valuable by stunting the plant's growth.
And we have Rosé, the ultimate genetic imposter, an entirely different plant carrying a Teolil and Pinot DNA that snuck into the royal court, mimicked the king, and was legally codified as an official biotype by sheer historical accident.
We then unpack the chemical deception utilized by the plant itself.
The color trick.
The genetic lack of stable malvidin molecules and the total reliance on unstable, highly reactive peonidin anthocyanins. This evolutionary quirk creates a pale, fragile, visually deceptive brick-red liquid that actively masks an iron fist of massive, mouth-drying polyphenolic tannins.
It's a visual trap set by a highly reactive plant that has to wrap itself in a thick, foggy, triterpenoid wax armor just to survive a grueling, high-risk growing season.
Which is why it won't travel.
Exactly. It clings to the specific marine soils of its ancient Italian homeland, refusing to travel, acting as a hypersensitive chemical translator of the dirt it grows in.
Which leaves us with a rather profound philosophical puzzle to consider, especially in an era where we prize scientific precision and genetic purity.
What's that?
Well, think about the facts we've uncovered. If a damaging, yield-restricting plant virus is directly responsible for creating one of the most highly prized and historically revered biological expressions in agriculture,
Right.
and if an entirely different genetic plant can be legally codified and protected as the original simply due to a centuries-old farming mistake, what does the concept of authenticity truly mean?
Oh, that's a great question. When we seek out products that boast of their pure, unadulterated heritage, what are we actually valuing? Are we valuing the pure DNA, or are we valuing the messy history?
Is the true magic of terroir, that highly sought-after sense of place found just as much in the local pathogens, the random genetic mutations, and the well-intentioned historical mistakes of the farmers, as it is in the climate and the soil itself?
It's absolutely something to think about the next time you find yourself looking for perfection or demanding a clean binary answer from nature. Because as we've seen today, when you step out of the sterile certainty of the X-ray machine and wade into the complex, mutating, virally infected reality of ancient agriculture.
Those diagnostic muddy waters.
Exactly. Those diagnostic muddy waters might just be where the true chaotic genius of nature actually lives.